Okay, so today's question was, what is the function of the dendrite, cell body, axon, and axon terminals? So dendrites receive incoming messages. Good. Cell body integrates and processes the messages. Good. Axon terminals can send information to other cells. Completely correct. Axons send messages away from the cell body. All right, very good. So we are going to continue today with our notes. So our notes in the last class were on really the structure of neurons. And then we talked about two different types of neurons, unipolar and multipolar neurons, and the different types of messages that they send to and from and throughout the body. Okay. So we're going to finish our notes here. These notes are accessible in Canvas, so if you just go back a couple module pages, you will find the notes. I am also sharing them on my screen. So in the last class period, we got through slide 12 here, and we are going to continue on and finish today. All right. Now remember, I do recommend, obviously you are going to do you and whatever works for you. But I generally recommend when we take notes that for most kids, you actually take out like a notebook and paper and hand write the notes as I go over them. Most students, if they just sit here and listen to me while I go over the notes, you really don't pay attention and you kind of zone out. At least when you're writing it helps you retain the information, but it also helps you process it so that it can help you realize, like, maybe I need to ask a question about this. I don't quite understand how she explained this, something along those lines. And if at any point as I'm going over the notes, you have a question about anything, so it could be just kind of a curiosity that is loosely related to what we're talking about, or it could be something that you need me to clarify or explain again, always drop it in the chat so that I can help you out, okay? So we are continuing here with our notes, and we're starting with the nerve impulse. So when we talk about these messages that our cells are sending to each other and sending to and from our brain or central nervous system, well, they're not really messages in the sense of like an email, a letter, a text message. A message in our body is actually a series of chemical events that trigger something else to happen. So if you ever in middle school or like 9th grade made like a Rube Goldberg device or machine where you kind of like stack all these things together and the action of one part of it triggers the action of another, well, that's very similar to how messaging and communication happens in our bodies and between our cells. So a nerve impulse is actually a chain reaction of chemical events. And these chain reaction of chemical events and reactions is what our body uses to pick up on stimuli or changes that are happening, and then to respond to them. So make something happen, regain balance, go back to normal, whatever the response might be. And then per usual, if you remember from the last class, I will awkwardly pause for a couple seconds here to make sure that anyone that is writing has enough time to catch up. Are everyone's classes going okay so far for second semester? I mean, it's only the second week, but. Wonderful, they sound great. I would believe that. For most kids, unless they're taking AP classes senior year, I would generally say this, or maybe a math class, depending on if they're in a higher level math, would be the hardest classes they're probably taking. All right, so then if we look at this picture here, let's see if there's a couple things we can recognize. All right, where the number 2 is here, I see a unipolar neuron, right? And I can tell it's unipolar because the cell body, this blob here, kind of seems detached from dendrites. And remember, that's because the unipolar neurons, their job is just to pick up on any stimuli or changes, send it to the brain as fast as possible, okay? And so we can see here, the stimulus was a phone rings, so they hear it. That sound is the stimulus then that gets sent through the unipolar neurons to the brain to tell the brain you heard a noise, okay? Sound then is based off memory, right? Our past experiences having heard it and what we associated with those sounds. So then based off previous memories and learned responses to those sounds, decision or response is made by the central nervous system, and it will be sent out neurons to make you answer the phone. Again, the neurons that send the message from the brain to your body part to make something happen or respond, those are the multipolar neurons. And I can recognize those because I've got the cell body directly attached to the dendrites. So again, when we talk about sending these messages again through these neurons, we're actually going to go in depth to talk about what chemical events or movement actually happens in these neurons to allow one change to be sent and notified to the brain and another decision to be made. So there are 5 basic steps to a nerve impulse. First, a neurotransmitter has to bind to the dendrites of a neuron. Hopefully you remember from the last class, a neurotransmitter is a chemical that is produced by our nerve cells. And these chemicals can stimulate a whole bunch of things to happen. They can make more nerve impulses or messages happen. They can make our organs and body parts do things and respond. After the neurotransmitter binds to the dendrites, we have depolarization and repolarization. The neuron releases new neurotransmitters, and then the neuron has to do something called regaining resting potential. Fadl, I could definitely see that. The beginning of a semester when, like, for the most part, right, you're easing into classes, way less stressful than the end of semester scramble to like redo things, retake things, submit late work, study for finals. End of semesters are always stressful. That's how I felt when I graduated high school. Get me the flock out of here. Agreed, Renato. Well, you don't have that long. This semester generally for seniors goes by shockingly fast. It'll get emotional. You know, I did not cry a tear when I graduated. Like real life, just get me out of here. I don't. Yeah, I don't know. It didn't seem sad to graduate high school to me. You know, people that are actually like true friends of yours, you will keep in touch with, and the people that you won't keep in touch with, realistically, you were probably never actually friends with. Just acquaintances that you saw in hallways and classes. So actually, Renato and Jennifer, the 10-year thing, students are the ones that have to organize class reunions. Schools don't organize class reunions. It's actually dependent on students. So if students don't organize one, there is no reunion. All right. So we are going to rewatch this video. You might remember we watched this video at the end of the last class. It's going to introduce us to some of the basic mechanisms that are going to happen in a nerve impulse. We're then going to look at a diagram to kind of summarize it, and then we'll get into the more detailed steps.
A nerve impulse is an electrical current that travels along dendrites or axons.
Oh, Lo siento. My bad.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
All right. So if we look at this diagram here, this is a diagram of a multipolar neuron we looked at in the last class. All right, so to orient yourself, starting from left to right, I have my dendrites. They look like the tree branches, and they receive the incoming messages that get integrated in the fried egg looking part, which is the cell body. The cell body then prioritizes those messages, and outgoing messages are sent through the axon here, this long cord, to the axon terminals. If the axon here is wrapped in something called a myelin sheath, that outgoing message or nerve impulse will be sent faster. And when it reaches the very end here, the axon terminals, a new neurotransmitter will be released, and either another nerve impulse will be stimulated or an organ will do something. So if we connect this now to what we're talking about with the nerve impulse, the neurotransmitter that first has to bind to the dendrites binds to the dendrites of the neuron that's going to send the message or nerve impulse here. Depolarization and repolarization then happens over and over again down the entire length of the axon until it reaches the terminals to stimulate the next action. So in order for us to talk about what happens along the axon of the neuron, which is really where the bulk of the nerve impulse happens, we have to talk about how neurons are set up before a nerve impulse. We use the term resting potential to refer to the natural state of a neuron, or how a neuron is set up, if you will, when it's doing nothing, when it's not completing a nerve impulse. Remember that our cells, which are neurons, one type of cell we have, are made of, on the most basic level, different types of molecules that are made of atoms. And so really, our whole existence is this beautiful and complicated interaction of atoms, nonstop, all the time.
Or just a giant blob of overly complex atoms.
We are. And in the case of neurons, we have more positively charged ions on the outside than the inside of a neuron. So an ion just refers to an atom that has a charge. In the case of a resting neuron, there are more potassium ions on the inside of the axon and more sodium ions on the outside of the axon. And there's actually way more sodium ions on the outside than potassium on the inside. So it makes the outside slightly more positively charged. You might remember from chemistry that whenever we have an imbalance of charge, we say that something is polar, kind of like with water molecules. So in this case, the axons of neurons are naturally polarized, meaning one side, the outside of the neuron axon, has a different charge than the inside. Now, things in nature don't like there to be an imbalance. We don't like there to be more energy in one place than another, right? We want the energy to spread out always. Particles always diffuse so that we don't have a higher concentration in one place or another. Same goes here with charges. It's unnatural to have this imbalance, but this imbalance is actually what's going to drive the nerve impulse.
And some people have a wrong amount of certain chemicals in their brains, and their body doesn't diffuse it as fast, and as a result they have a condition known as depression.
100%. So those, that have clinical depression or clinical anxiety, the clinical in front means that physiologically speaking, their DNA does not actually code to produce the proper amount of chemicals. And those chemicals actually that Cassie's talking about are neurotransmitters. Dopamine and serotonin, which are the common neurotransmitters associated with imbalances that could cause anxiety or depression. It's saying that their body simply does not have the directions or blueprint to produce them at all or to produce their correct quantities. And so it is difficult or impossible for them, depending on the degree of the clinical diagnosis, to interact, socialize, and respond to things the way others might who have proper chemicals. So if we look at this diagram here, kind of my red bars represent the axon. So think of the axon like this giant tube, okay? On the inside of our tube here, on the inside of the axon, I have potassium ions naturally. And on the outsides of my axon, I have sodium ions naturally. I have more sodium on the outside than potassium on the inside. So this imbalance, this unequal concentration of charges is going to drive the movement of these charged particles in a nerve impulse. So after the neurotransmitter has stuck to the dendrites, now we move on to depolarization and repolarization. Depolarization and repolarization is really the bulk of the events that happens in a nerve impulse. And it happens over and over again down the length of the axon of the neuron. When we depolarize something, we are removing the uneven charge or the imbalance of the charged particles. So during depolarization, the sodium pumps open and the sodium ions move into the axon. When we repolarize something, we bring back that uneven charge or imbalance of charged particles. During repolarization, the potassium pumps open and the potassium ions move out of the axon. What ends up happening then is this series. So again, if we envision that our axon is this big kind of like tube or highway here, and we've got some particles on the outside and some on the inside, what ends up happening is this series of almost what looks like maybe like a dolphin jumping in and out of the water. So in depolarization, I have sodium ions going in, repolarization, potassium out. Depolarization, sodium in, repolarization, sodium out. This happens down the entire length of the axon. That is a nerve impulse. So when we talk about these messages being sent to and from cells, we're talking about this movement of charged particles and the release of a chemical at the end. Think back too, right? So some of you might also be thinking, well, I thought, maybe our brain runs on something like electricity. We see them monitor that in the hospital. We can use electricity to re-stimulate organs. Well, that would be correct because this is an electrical current, right? Think back to physics and physical science. What is electricity? It's the movement of charged particles. And that's what we have here. So you essentially have an electrical current that travels down the length of your axon to stimulate something else to happen. All right, I will pause for a second. And as you are kind of processing what I'm saying, again, if you have a question about something I'm saying, or maybe it makes you think of something else that you're curious about, do not hesitate to drop it in the chat. All right, does anyone need more time or have a question they want to ask? I'll take that as a no. All right, so again, remember in this diagram here, these kind of red bars represent the axon, like a tube. And so we have like the inside of the tube or the axon and the outside of the tube or the axon. So in depolarization, we have sodium ions move in. So you can see this blue arrow shows here, these sodium ions that were on the outside move inside the axon. In repolarization, the potassium ions that were on the inside move out of the axon. We have this then repetition happening all the way down the length of the entire axon, in and out constantly. Now, once depolarization and repolarization reaches the end of the axon terminals, a neurotransmitter will be released. Neurotransmitters, again, are very specific to body parts and what they are used for. Some, like Cassie mentioned, are used to stimulate emotional memories and responses. Some are used to stimulate digestion or muscle contractions. So it just depends on what response the body requires. This neurotransmitter then is going to bind to either another neuron and stimulate another nerve impulse, or it will bind to an organ and make the organ do something. And again, that could mean it binds to your heart tissue, the cells in your heart tissue to make your heart beat. It could bind to cells in your lungs to make them inhale or exhale. It could bind to cells in the duodenum of your small intestine to make it release a bile or pancreatic juice. And then I'll pause again. All right, so let's look at, what happens here after this point. So in this picture here, we kind of see we zoomed in on a neuron. So I see my multipolar neurons here at the top, and we zoomed in at the axon terminals. So we're saying again that that depolarization and repolarization event reached all the way to the axon terminals. These chemicals, so the little red dots and the bubbles here, are released from the terminals, and they're going to stick to or bind to receptors on the other side. And again, the other side could be the dendrites of the next neuron because we have to send more messages, or it could be receptors on a muscle tissue to make the muscle contract, whatever it might be. All right, we're going to watch another video to kind of hopefully summarize some of this. And again, as you watch it, if you have any questions, do not hesitate to drop them in the chat.
Hi, it's Paul Anderson, and in this video, I'm going to go over the basics of the neuron. The neuron is the basic unit in the nervous system. In a simple animal like a nematode worm, they only have 302 neurons, but what they allow them to do is to take information in from their surroundings and respond to that information. If you look at how those 302 neurons are connected, it's very complex. Now, humans are quite a bit bigger, and we have 100 billion neurons in our body. Most of those are centered in the central nervous system, in the brain, and the spinal cord, but the basic unit of our nervous system is the same. It's the neuron. If we look at the neuron anatomy and remove all the other neurons, the center is going to be what's called the cell body. This is a typical cell. It's a metabolizing cell. It's taking in energy. It's using that energy, and so it's going to have a nucleus. It's going to have Golgi apparatus, mitochondria. It's a typical cell, but there are extensions of those cell bodies called the dendrites. Those dendrites are connected to other neurons, and they're taking information in the form of neurotransmitters. In fact, this whole side of the neuron is receiving information. We then have the axon. The axon is where we're transmitting information. It begins at the axon hillock and is going to move to the terminals. Now remember that the terminals, there's a synapse that connects us to other neurons, but the signal is going to move in this direction. We're taking in information from the dendrites, the cell body, and then we can send information to the next neuron. Now, how do we do that is through an action potential, and I have a video that I'll put a link to where I go into the specifics of how that occurs. But certain axons in our body are really, really long, and for that information to travel quickly, we will wrap them in myelin sheaths. It's a Schwann cell that's wrapped around it. The way it works is we put the voltage-gated channels in between that, and it acts to insulate that nerve. It allows the information to travel more readily down that axon. And we generally say if you have an axon that's wrapped in myelin sheath, we call that a nerve fiber inside our body. Now we're taking in information as well. So there's going to be another neuron that's connected to both the cell body and the dendrites of that cell. But there's going to be a gap between these two, and we call that a synapse. It's a connection between neurons. Again, I have another video on the synapse if you want to dig into the specifics, especially of how chemical synapses work. Neurons are classified in two ways. The first way is due to their structural classification. So if we take the cell body of a typical neuron, like this multipolar neuron, the way you can figure out what type of a neuron is find the cell body and figure out how many poles are coming off of that cell body. So if we count them, we have a number of different dendrites that are coming off of this neuron. And then we have one axon that's coming off. And so we call that a multipolar neuron. This is the most common neuron that we have in our body. If we look at the next one, we find the cell body again. How many poles do we have coming off of it? We've got the axon coming out and the dendrite coming in. And so we call this a bipolar neuron. Bipolar neurons are very rare. They transmit information from the receptors in our eye, receptors in our nose. If we look at the next one, here's the cell body. You can see there's only one pole coming off of that. It's actually a fused axon and dendrite, and we call that a unipolar neuron. Now, in vertebrates, it's sometimes referred to as the pseudo-unipolar neuron. These are going to be most of the sensory neurons in our body. They're taking information in and transmitting that back into the central nervous system. And then if we look at the next one, if we look at that, you can see there's no axon on it at all. And so we call that an anaxonic neuron. These are found in the brain, and they're not super understood. Now, there are a lot of other structural classifications, but these are the general ones that you're going to find in a typical anatomy class. Now, neurons can also be classified according to their functional classification. Those that take in information, stimulus coming in from our environment, and then the response that we have. So if we kind of move that to the central nervous system, we're looking at a cross section of the spine. So we're taking information in from receptors. So these could be in the skin, for example, touch receptors, heat receptors, pain receptors. And so we have to transmit that information to the central nervous system. So those neurons that bring information in are sometimes referred to as sensory neurons or afferent neurons. You can see this is a unipolar neuron, and it's transmitting that information into the central nervous system. And then we have information going back out. We're sending information out to effectors in our body. It could be muscles, could be glands, and we're going to call those motor neurons. They're moving away from the center. Sometimes we refer to those as afferent neurons as well. Now, there should be connections between those neurons. Those aren't all the neurons we have in our body. And so we also have these connections in the middle, and those are going to be called interneurons. And so that's the neuron. How did you do? Can you remember the different parts of the neuron? Do you remember that these are the dendrites, the cell body? Do you remember what type of a neuron this is? If not, go back and rewind it. But those are neurons. Those are the basics, and I hope that was helpful.
All right. So after a whole nerve impulse has happened, the neuron actually has to go back to how it was before this all happened, and we call that regaining resting potential. So that means that we have to force the sodium ions back out of the axon, force the potassium ions back in, so that everything is set up just like it was before the nerve impulse happens. Because we have to force these ions back in and out, it does require a little bit of ATP, which is an energy storing molecule in our cells. But again, it's essential. So it's like, I just want you to take a moment to think and appreciate of how insane this process is that is constantly happening, right? Every single unique thought you're having in your head as I'm talking while you're also listening to me, looking at me, you're also writing down the notes here that are projected on the screen. And then writing the notes also requires that you have hundreds of different muscles in your hand, arm, shoulders, back that are all functioning properly to allow you to write while you're sitting upright, which requires different muscle engagement. We're talking millions to trillions of nerve impulses nonstop all the time for these very simple basic things. And they happen so quickly, right? For me to just move my finger takes nothing but a second. But we are saying that these nerve impulses, the neurotransmitter binding, depolarization, repolarization, depolarization, repolarization, down the length of the axon, the release of the neurotransmitter, that has to happen multiple times for the simplest of things, okay? And so it's just impressive how efficient our body is, and then to have to put it all back where it was before I can move my finger in a different direction in another way. Well, that's insane. It's at least insane and impressive to me. So if we look here again at the end of a nerve impulse, we had sodium mostly on the inside, potassium mostly on the outside. We have to force them back in the opposite direction so that we can set our neuron back up for another nerve impulse. Okay. And so here, this video, I thought visually did a really good job of showing what's happening down the axon.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
All right. So the last little nugget we have to cover here is the myelin sheath. So on Monday, I said that the myelin sheath covers the axon, and it helps increase the rate of messages being sent or speeds them up. Well, that's because the myelin sheath actually covers or wraps around some of the sodium and potassium channels. So I always compare an axon with a myelin sheath to a freeway versus an axon without a myelin sheath, like main roads and side roads. On main roads and side roads, you know, every so many feet, you have to stop at a stop sign, you have to stop at a yield sign, you have to stop at a stoplight. It's constantly go a little bit, stop, go a little bit, stop. Well, that's what it's like in an axon that doesn't have a myelin sheath. These sodium and potassium ions have to go in and out every little micrometer down the length of the axon. Will it still happen? Yeah. Does it still happen fast? Yes. But it could happen faster. Just like driving could happen faster if you took the freeway. When you take a freeway right, you get on the on-ramp, you fly down the length of the freeway until your destination, and you get off the exit. Well, ions can do the same thing when an axon is wrapped in a myelin sheath. They can enter where they're supposed to, fly down the length of the axon, and exit back out the next opening. It simply allows the process to happen much faster because there's not as much stop and go along the way. So I can come back to this slide real quick. Let me just kind of show you here in this diagram. So again, imagine this yellow tube here is our axon. And that axon is full of these channels that allow the sodium and potassium ions to move in and out. Well, if I wrap around it this fatty tissue called the myelin sheath, I'm inevitably covering some of those channels. And it doesn't cause a problem. Again, what that's actually doing is preventing the ions from going in and out every little bit down the length of the axon. Instead, they go in and they fly down to the next opening and then they go out. And so it speeds up that process. So we're gonna watch one more video.
Saltatory conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier, which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction. And it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
All right. So over these 2 days, then we covered all the structural parts of a neuron and how neurons send messages throughout our body or nerve impulses. Your job now is to work on your 1.10 neurons and nerve impulse practice assignment. This practice assignment is going to be due this Sunday. I'm having it due this Sunday just because it is a little bit lengthier. I wouldn't say it's difficult, but it is a little bit lengthier, and I know some of you work and whatnot. So if you finish it before Sunday, turn it in. But if you need to take till Sunday, that's perfectly fine. In this assignment, part 1 is just basic vocab and concept review. So you have a little crossword puzzle to provide hints. Then the questions for the crossword puzzle and a place for you to type the answers are in the tables below. So I can actually just type whatever my answer is there. Following that, in part 2, we have some diagrams. So these are diagrams just like we looked at in our notes, and it's just to help you practice what the different parts of neurons are, what their functions are, differentiating between unipolar and multipolar neurons, and then what's happening during a nerve impulse, okay? So those diagrams will also be helpful if you understand them when we get to our models. So you see it does go to 55. These answers, a lot of them are one-word answers, so it's not that it's difficult or lengthy as in typing or writing, but I do just know a lot of you have jobs, so if you need till Sunday, that's fine. If you finish again early, submit it early. This is your assignment that you're going to work on for the remainder of class today, and again, it is due Sunday night. Does anyone have a question about this assignment or anything else? Okay. So if you would prefer to exit Zoom to work on your homework, you are more than welcome to do that. Remember, your next class starts